Welcome to Mr. Chalk's Revision Tips. In this video, we will look at the stages of mitosis and the Mitotic Index. You might notice as you're working through your PowerPoint, there will be certain checkpoint questions that you need to have a go at as you get to them. Mitosis is part of the cell cycle or part of the cells where it divides into two. So remember, a chromosome is the DNA part of the molecule. The stages of mitosis are prophase, metaphase, anaphase and telophase, and the mitotic index is defined as the ratio between the number of cells in a population undergoing mitosis to the total number of cells in that population. Mitosis is part of the cell cycle when replication or replicated chromosomes is separated into two new nuclei. Cell division gives rise to genetically identical cells in which a number of chromosomes are maintained. So if you start off with 46 chromosomes, you end up with 46 chromosomes. Before mitosis can occur, all of the DNA needs to be replicated during interphase. Each DNA molecule is converted into two identical DNA molecules called chromatids. The two chromatids are joined together at a region called the centromere. It is necessary to keep the chromatids together during mitosis so that they can be precisely manoeuvred and segregated equally, one to each new daughter cell. During prophase, chromatin fibers begin to coil and condense to form chromosomes that will take up stains and become visible under a microscope. The nucleolus, so a distinct area of the nucleus responsible for RNA synthesis, disappears and the nuclear membrane begins to break down. Protein microtubules form spindle-shaped structures linking to the poles of the cell. The fibers forming the spindles are necessary to move the chromosomes into the correct position before dividing. In animal cells and some plant cells, two centrioles migrate to opposite poles of the cell. The centrioles are cylindrical bundles of proteins that help the formation of the spindles. So the spindle fibers attach to specific areas on the centromeres and start to move the chromosomes to the centre of the cell. By the end of the prophase, the nuclear envelope has disappeared. During the metaphase, the chromosomes are moved by spindle fibers to form a plane along the centre of the cell, so they all line up in the middle. So the metaphase plate is held in a certain position along the equator of the cell. Anaphase is a stage of mitosis and meiosis where genetic information is moved to opposite poles of the dividing cell. There are two stages of anaphase in mitosis. Anaphase A, where the chromosomes move to the poles, which is followed by anaphase B, where the poles kind of move apart. A characteristic V shape of the chromatids can be seen as they are pulled through the cytoplasm of the cell. In telophase, the chromatids have reached poles and are now called chromosomes. The two new sets of chromosomes assemble at each pole and a nuclear envelope reforms around each set. So this allows cytokinesis to begin. And finally, cytokinesis is the physical dividing of the cell into two genetically identical daughter cells. So the mitotic index So when mitosis is actively occurring in a tissue, such as root tips or wherever, there's a certain amount of cells that will be dividing and a certain amount of cells that won't be. So to work out the mitotic index, you use the equation, the number of cells with condensed chromosomes, so they are the number of cells in which under a microscope you can see the chromosome, and you divide that by the total number of cells. So for instance, if we have 29 cells in the sample, if we can clearly see 3 cells or the chromosomes in 3 cells, you would divide 3 by 29, then times that by 100, which would give you the percentage of the cells that were undergoing mitosis at that time, and that would be your mitotic index.
Thanks for watching.